Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you. Join over 350,000 people just like you who are taking control of their wellness journey with Viome. When it comes to choosing the right food and supplements for you, don't guess, test. With Viome's Health Intelligence Test, you get over 30 health insights based on your unique biology and your gut microbiome. You also receive personalized food recommendations and precision supplements formulated literally just for you. Use code GENIUS to get an extra $20 off a health intelligence test. Visit Viome.com to shop now.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. I have a returning guest, Brendon Coventry. He's an associate professor of surgery in Adelaide, South Australia. We're going to talk about a vaccine, actually, from melanoma that he's been working on and researching. So, Brendon, welcome back. How are you doing?
Yeah, thanks a lot, Richard.
In case this is listeners' first listen of you, just give me a little bit of your background, and then let's talk about your current work.
Yeah. Oh, look, thanks. It's a bit unusual to have a surgeon playing around with vaccines and immunology, but I did a PhD in immunology some years ago, about 30 years ago, and I've been interested in immunology really during that and ever since, and it's pretty fascinating. So my real training is in PhD immunology and then surgery, and my day job's as a surgeon, but I do a lot of research in immunology, particularly vaccines, looking at trying to boost the body's own immune system against cancer cells and trying to remove cancer cells. And we've had a modicum of success in that area.
Okay, so is this this is a vaccine? But I mean, vaccines usually, from what I understand, they happen before a person gets sick with, whatever it may be, viral bacterial infection. This sounds like a post, obviously, if you can call it cancer an infection, but this is a post intervention. It seems like. So
yeah,
is it proper to call it a vaccine or a treatment?
Yeah, absolutely. You've really hit on it there, Richard, in that you're right. Many people, just about everyone, thinks of vaccines as being the type of thing that you give to like a part of an infection or some component related to the infection that can actually prevent the actual infection itself and the severe infection occurring. And that's kind of what we're trying to do with COVID, we're trying to do with measles, polio, and many other diseases that we get vaccinated against. So that's sort of like a conventional view of vaccination. But what we're talking about here is something a little bit, it's very similar, but it's something a little bit different. And that is that when you've got an established cancer growing, itself is inciting some form at various levels of immune reaction. It's very rare to have a cancer that doesn't incite some level of immune reaction. So the body's aware of this cancer sitting there growing, and there's tiny little molecules, or complex molecules, on the surface of cancer cells. In fact, they're on the surface of a lot of body cells, and these are given off as the cell dies or as the cell's damaged, and the body's constantly reacting to these. So the immune system's picking up these molecules and causing a reaction, and that reaction is capable of slowing the cells in terms of their growth, or even killing the cells. So parts of the immune system can attach to the cancer cells and blow them apart, basically. And it does this through a variety of mechanisms, and we're really just starting to uncover some of those mechanisms. And it's capable of killing tumour cells, and it's capable of killing whole masses of tumour cells. In fact, in some cases, kilos of tumour cell. So that's sort of the principle of vaccination, used not as a prevention, but as a treatment for cancer.
What is the mechanism of action, without giving away proprietary info, but how does this treatment work to help kill cancer cells?
So people have been playing around with a lot of vaccines over the years. In fact, it goes back more than 100 years, where people have used parts of infectious molecules, various ways of trying to vaccinate against cancer, and been moderately successful. And then there's some ways that should work that don't seem to be very effective at all, and that's kind of coloured a lot of people's views of vaccines as a possible form of cancer treatment. So there were a lot of trials done around the 1950s, for example, 60s, 70s, and into the 1980s. And what they did was to try and use cancer cells that had been immobilised in some way, irradiated, for example, and they could be injected into other patients. So cancers from one patient, irradiated to stop them dividing or causing any trouble, could be injected into other patients. And so there was a lot of work done there. There was cells that were split apart, and I'll talk a bit more about that in a minute, because that's turning out to be one of the most important ways. So cells could be split apart, and they could be used to create vaccines, either from the patient's own cancer or from someone else's cancer, and that could be used as a vaccine to try and treat the cancer. And then the thought was that maybe if there's a component of these, some tiny little molecule, then maybe we could strip out that molecule and we could refine it, and then use those molecules as a much more specific way of treating. And it turns out that the more simple you make the vaccine, so if you do strip out these molecules and use them to vaccinate, it seems to become less effective. There's some problem with actually simplification. So you would have thought that maybe if it was purer and closer to the sort of thing that was causing the reaction, then maybe it should be better. But that's not the way it's working out. So vaccination is the mechanism that is working here is it's never been particularly clear. And I think this is another thing that's coloured people's views of things. You know, it's nice to have a simple drug molecule that you think is working a certain way, and believe it or not, we're finding out that a lot of them aren't working that way. They're actually working different ways to the ways that they're actually registered for, and or they have some extra effects, say. So we, in terms of the mechanism of action, we're still unpicking that. We're still trying to work it out.
what do you observe under, I mean, are you able to observe any action under light microscope in a petri dish or anything, or can you use an organoid model of this cancer? Like, you must be able to see some kind of action.
Yeah, you can. So the sort of action that you see is an incitement, if you like, or a boosting of the immune system reaction. So you can measure that by looking at the activity of cells that come from the white cells that come from the blood, and they can be activated. So you can measure various levels. It's not easy to measure terribly much in the immune system that's really reliable, but you can measure things that change as a result of this vaccination process occurring. For example, with a lot of our vaccines that we're talking about recently with COVID and other things, you can measure antibody responses. And with just like that, you can measure antibody responses to vaccines against cancers as well. So you can measure things to do with the cells, the cellular responses, and you can measure things to do with the release of chemicals like antibodies, which is the humoral responses. You can also measure the release of certain products from the cells that boost immune system function, like the interleukins, for example, which are very important for boosting and growing, expanding T cells, for example, which are part of the white cell population, and very important for fighting cancer. So yeah, you can measure lots of components that show that there's a reaction going on and a boosting going on, but a lot of them aren't terribly reliable as biomarkers to predict whether a particular patient's going to get the response or a patient might not. So it's a little bit fuzzy around the edges, if you like, and it's not as if you're measuring something in the blood like an element like lithium or glucose or something like that in the blood, and you can say, look, this is going up. This shows a definite change. A lot of the immune system measurements are a little bit less clear than that, and so you have trouble really showing exactly why immune responses are occurring effectively in some people, but not effectively in others. You can see it.
How do you characterize the immune response? It's, the generic words immune response don't really tell me specifically what's happening. Like, what is happening?
So the way we know it's working, for example, is that the tumour is regressing and in some cases completely disappearing. So we've got really good evidence. We've got a number of patients that are alive from anywhere between 15 and 22 years that have been vaccinated against their cancer. So the cancer is busy growing, and you come along with your vaccine and you inject, and it usually requires a number of different vaccinations over a period of time, and the tumour just disappears. It just goes. So that clearly the vaccine has caused or incited something that is been going on in the patient, maybe at a lower level, maybe partly switched off and not activated terribly effectively, and it turns this around and activates it effectively so that it can cause the immune response to effectively injure the tumour and remove it. So you can see the clinical responses, and you know that this is something that's occurred through the immune system. The immune system's a mediator here, but trying to actually measure the exact components that are going up and going down that are causing the response is a bit more problematic. And there's a lot of work going on.
But how would you have developed this or contemplated in the first place if there was no proposed mechanism of action?
Well, I mean, how do we contemplate or propose most things? A lot of our scientific discoveries and things that end up as treatments are just totally serendipitous. So we might observe something. So, for example, when you give a treatment that damages a cancer, for example, radiation treatment or chemotherapy, cytotoxic chemotherapy, you can actually observe the damage. So if you take biopsies, or you see a part of the tumour it's able to be observed well, then you can actually see the damage that's occurred as a result of those treatments. And it's that damage that actually produces a vaccine response as well. So we've known about this for a while, and what it does is it releases little parts of the molecules that are on the cells, and the cells can die as a result of the therapies, and they actually, you get a double whammy. So not only are the cells killed, but the cells release products, and those products actually act to vaccinate the patient, because vaccination's a natural, continuous process that's going on all the time in all of us, and it's just completely under the radar. It's very hard to measure, it's very hard to see, it's invisible, but it's going on all the time. So we're constantly getting exposed to environmental chemicals. They come from in the form of allergens and various other things in the air that we breathe, absorbed through the skin. They can be eaten, and then the gut takes them up, and they get into the system.
It just seems like a misnomer. This doesn't seem really like a vaccine at all. It just seems like immunotherapy. I just wonder why would you want to call it a vaccine instead of immunotherapy? Is it easier to get it out there, or is there more interest because it's called a vaccine, or why?
No. In fact, it's a vaccine like immunotherapy. The term immunotherapy is only a fairly recent invention. It's immunotherapy is just an immune system therapy, and this is an immunotherapy. It's just an immune system therapy. But what we've been working on is something that actually turns the immune system around. Now, you can argue that a lot of the immunotherapies that have just been developed fairly recently for cancer treatment, like the anti-programmed death 1 receptors, anti-PD-1, trade names are pembrolizumab and nivolumab and things like that, and the anti-CTLA-4 therapies like ipilimumab, for example, they're sort of recent immunotherapies. A bit earlier to that was something called interleukin-2, which expands T cells and boosts T cell responses. So prior to that was tuberculosis vaccine, and that was used as an immunotherapy as well. And that appears to work by, and we still don't know, but it appears to work by boosting the innate immune system, or the very primitive immune system first, and then that ends up inciting the more complex immune system, or the so-called adaptive immune system, and causes an effect not against the tuberculosis bacterium, but against the tumour itself. So that's used, for example, in bladder cancer. So immunotherapy is a broad term that's used, and this is a type of immunotherapy. Why it's a vaccine, though, is that you give a little component of it, and it's able to get a big response. And that's what we do, if you think, with the preventive vaccines. So we use those, and what we do is we inject a tiny amount of the polio virus, which can be live and attenuated or made weaker, or it can be killed. So that some vaccines are killed vaccines. Interestingly enough, the live vaccines are generally stronger and longer lived. And one of our components of our vaccine is we use a virus, and it's the old virus that was used against smallpox, and vaccinia virus, and we can actually boost the immune response using vaccinia virus. Other viruses will do it too, but vaccinia virus is really neat because it actually can lyse cells or split apart cells as well.
Are you using it as a vector to get a different payload into the cells, or how are you using it?
Yeah, well, it's interesting you say that, because we're not, but other groups are trying to use viruses as vectors. So things like adenovirus and vaccinia virus, they can use them as ways to sort of transport it into cells. So if we know it gets into a cell, we can actually use the virus as to add something to and take genetic material or protein or whatever into the cell. So that's the purpose of a vector. But but in our system, we're not using it that way. We're using it as a way of actually damaging the cells and splitting the cells apart and creating the vaccine. So it's a really neat way of getting a double whammy, because you get parts of tumour cell and you get virus, and the virus can actually stimulate the immune system. We've always known for a long time, Richard, that the immune system can be stimulated by viral infection, and you know that's the reason that people get into trouble. It often overstimulates the immune system. For example, in COVID, most recently, but also in conditions like hepatitis, where the liver gets overstimulated and the immune system starts to attack the liver. We can have situations, many situations where the virus actually can incite an immune response and cause a massive outpouring of products from the immune system, which can overwhelm the system and cause the whole system to collapse. So it can be really dramatic sometimes. Sometimes it's a lot milder, like a common cold or a mild flu type infection, where there's a reaction for a period, might be mainly respiratory, and then there's a recovery and it's overcome. So we know viruses can incite immune responses, but really interesting is the fact that people can sometimes get infections, and these can be bacterial infections or viral infections, so completely what you'd imagine are completely different infections, and get rid of their tumour as a part of having this infection. So infections are able to gear up the immune system against infection, but the side effect is that you actually gear up the immune response that's already going on against the cancer as well. And occasionally these infections can get rid of cancer altogether. So we know that infections are a very powerful way of boosting the immune response. And in a way, a lot of these newer immunotherapy treatments do exactly that. What they do is they just drive on the immune system, fire it up in a non-specific way. And as a side effect, you get the activity of the immune system against the cancer being boosted.
Right, but again, what was if you don't know the method of action, what was the serendipity that caused you to be able to develop this, to find it? There's got to be some way. It wasn't just random.
No, absolutely. And so there was a lot of work going on in around the 1950s, when the people started to get interested in maybe you could use cancer to vaccinate against cancer. And so by this I mean that people started to realise that you could actually break down, and you can do this a whole variety of different ways, try and kill cancer cells in the lab, and then you could take those cancer cells and inject them back into the same patient, and that you get an immune response occurring. You could test them against the white cells that you could take from that same patient in the lab, and you could test them in the lab and see if the white cells were stimulated. And the observation was being frequently made that yes, you could actually see reactions of the body's own white cells against the cancer cells that were broken down, and so you could demonstrate this. And then people started to look at this a bit more, and this was quite a number of labs across the world, all sorts of different ways of manufacturing cancer vaccines this way. And so that's probably why it got the term vaccine early in the piece. And then the immune system could be stimulated, taking part even better by taking parts of someone else's cancer, splitting it apart so it couldn't divide or cause any trouble, and then using that as a vaccine to inject into people with a similar type of cancer. So this was. Are
you sure you're not affecting the localized microbiome of the given tumor and influencing that way? Could that be a method of action, not just immune stimulation?
Yeah, we probably understand less about the microbiome of the actual tumor itself than than the microbiome anywhere else. And we don't understand a massive amount about the microbiome in a lot of other areas. The main topic that's being worked on at the moment is the microbiome in the gut and its influence on sort of systemic immune responses elsewhere in the body, away from the gut. But in terms of the microbiome in the cancer, most of the cancers pretty much sterile in that, or relatively sterile in that there's not a lot of bacteria that are present inside the tumour. Now, a gut tumour may be a bit different. A respiratory tumour is a bit different because these are on the surface, so they're actually exposed to bacterial populations. But inside the body, in a deep part of the body, there's very few bacteria. There's not much of a microbiome that we can identify yet.
Discover how your gut microbiome is impacting your cellular health, immune health, and how you're aging from the inside out with Viome's Health Intelligence Test. Collect your samples, send them to the Viome lab, and within 2 to 3 weeks, your health scores and food and supplement recommendations will be available to you right in your Viome app. Visit Viome.com and use code GENIUS to get an extra $20 off your health intelligence test.
Just one thing that may be of interest to you is I had interviewed a scientist named Florencia McAllister, and they did find in pancreatic tumors there was a different localized microbiome than the rest of the pancreas, but they also did find a localized microbiome specific to the pancreas as well. So perhaps that assumption is not valid, and there is a microbiome that may be substantial enough in all parts of the body to affect, cellular action, change the trading of metabolites, bring along associated phages that may be protective to the whole arrangement for that particular cell and change its interaction with the rest of the body.
Yeah, well, that's possible. You see, the pancreas itself, I'm not familiar with that work, so you'd have to sort of look at this in a bit more detail. But the pancreas itself is part of the gut, so it actually has a duct system, and those ducts are actually exposed to the gastrointestinal bacteria as well to some degree. So if you're talking about ductal cancer of the pancreas, then maybe. But inside the pancreas itself, more remote from the ducts, maybe I wonder if there's a microbiome there, because you wouldn't expect one. And most of the tumours that I've been dealing with are inside the body, like within the breast. Although, having said that, the breast actually has its own ducts as well. So, maybe this is something that can be clarified a little more with more research into it. And it's not to say that bacteria and viruses don't exist within tumours. We know that they can and do in certain tumours, but it's something that I don't think we've really appreciated much, and so I can't really comment any further on that.
Yeah, that's okay. It's just something to point out that may be important. That's why I just again, no one knows, but it might be something to include in the picture of what's going on. When you guys are treating melanoma, are you treating it in the metastatic state? I mean, it seems like melanoma is incredibly prone to metastases. So even if it doesn't appear to be any, maybe it's in a metastatic state. And, when does your application work better versus not work as well? Does it work better in early stage melanoma or late?
It's been really hard to get a handle on that. It's a good question, though, because what we have noticed is that even patients that have extensive metastases, big metastases, and those that are seemingly, they've had other treatments as well, they're seemingly incredibly difficult to treat. They can sometimes respond incredibly well. Others that you might think have only got one or two metastases, you might say, well, gee, that looks a much earlier stage and it should be able to be treated a lot more easily. Don't seem to respond as well. So it's a mixed bag, and we don't have a really good handle. It's exactly the same as what you see with almost all forms of cancer treatment. For example, chemotherapy of many different types, immunotherapies of many different types, radiation treatment given many different ways and in many sites. It's very hard to predict who's going to respond and who won't. Situations that you expect might be much more easily treated and ones that you expect are incredibly difficult to treat. So, it's not straightforward, and it's something that we are struggling to get a handle on. You might imagine that there'd be some patterns though, Richard, and there are a few, but they're a lot fewer than you'd imagine. And
what are the patterns and what do they suggest?
Well, for example, we know that tumours in lung, for example, and subcutaneous metastases from melanoma appear to be easier to treat with immunotherapies than those that are related to bone and to some degree the central nervous system. So it's not necessarily site specific quite as easily as that, but it is to some degree site specific. So there's been a fair bit of work with this, and it really goes back to your question about the microbiome in a way, because there's a group in Canada that have been looking at different bacteria that are present in different locations in the body. So, for example, the lung and the skin, just two examples, have a different microbiome mix. And so their argument is that if you use a vaccine that can boost the immune system, you really need to adjust for this difference in the microbiome. And you might choose one type of bacteria for treating lung and another type of bacteria or a variation of it for treating skin metastases. So this is, or even primaries in those locations. So this is, there's some fascinating information that we've got that might have relevance, but it's been pretty hard joining the dots here, and this lack of pattern between the different treatments has been incredibly frustrating, and it's hard to see why there's not more of a pattern that's emerged, because you'd expect perhaps that intuitively that there might be some more common ground than we're finding. It's more haphazard than we've been expecting, and the reason for that could lie in the fact that when tumours grow, they don't grow as a single, like a tissue. They grow as tissues grow as uniform groups of cells. In cancers, they tend to develop genetic mutations and end up as a whole lot of different clones, so that they're not actually one thing. When we look at them from the outside as a mass, we imagine them as one entity, like a tissue, we say. But they're not quite like that. They have a lot of heterogeneity in them genetically and also in the protein structure as well. So this may explain why we get this sort of mixed picture, and parts of the tumour can actually respond to treatment and the other parts might not, and might continue.
Yeah, I know tumors are very heterogeneous, right? So I understand tumors are very heterogeneous. So are you able to observe that this treatment affects the outer part of the tumor, the inner part? Does it make it into a Swiss cheese, then the whole thing dies, or like what happens?
No, see, the interesting thing is that when you see the spectacular responses, you get complete regression. It all goes, the whole lot goes. We've had kilos of tumour just disappear as a result of this vaccination immunotherapy approach. So when the immune system's turned on, it's turned on in a really big and effective way. And this is probably our biggest glimpse into the actual mechanisms that will ultimately find and will ultimately be able to use in therapeutics for cancer. You know, the window's just a little bit open and we can see a tiny view through it, but we can't see the whole picture yet. And one of the things that it's very exciting because we actually know that there are treatments there. We know how to make it. We know we can make it effective in some situations, but we don't exactly know how to make it effective in more situations. And that's really the topic that we're drilling down on now, to try and see if we know we can get these responses. We know they're long term. We also are hooked up on this word cure too, because what is a cure? A cure presumably is that you don't die of that particular disease, and you can get rid of it, and maybe you can.
I think a cure would be that the disease appears to be, you're asymptomatic, it's gone, and you don't need any drugs to keep you going. Management seems to be the flavor of the decade or the century where, it may not kill you, but you still have to take X number of drugs in order to manage it and keep it going. So,
yeah, I don't know about that because if you talk to a lot of patients about this, they actually want it gone altogether. This idea of having a bit of tumour sitting there that's indolent and stable and not moving, and they're still busy living their lives, is they don't feel that they're cured. Certainly the patients I talk to don't feel that they're cured. They're reprieved and they're alive and they're not terribly troubled by it, but it's still there, and they're always frightened that it's going to come back. But it comes back to what I was trying to get at a minute ago, that this idea of the notion of cure. I had when the vaccine hit the press fairly recently here in Australia, and I've had calls from all over the place, all over the world, all over Australia, and people emailing me wanting for all sorts of types of tumours too, because there's clearly a massive need with people with cancer that have failed treatments. And so the stuff that we're throwing at cancers are working in a small percentage of patients in a lot of tumours, but they're not working for the vast majority of patients. So in the vast majority of tumours, and so what we've got is this massive area of unmet need, absolutely massive, and people are failing treatments every day. They're staring down death, and they really want something else. They're also terribly troubled by the side effects. The side effects of a lot of these treatments are horrendous. Some of them even include death itself as a side effect of treatment-related toxicity. But, there's a massive area of need. And one of the emails that came through was fascinating, because what it said was that from a daughter of a lady who had died only just a year or so ago, and 60 years previously she'd had a melanoma removed, and she was fine for 60 years until her husband died, and she became incredibly depressed and very, very upset, and she suddenly noticed that she had some lumps appearing. And those lumps, when they were biopsied, showed melanoma. To her knowledge and the knowledge of all the medical specialists, she'd never had another melanoma removed. So the question really remained as to whether this was a tumour that had been sitting indolent for 60 years and had finally become activated when the immune system had lost its guard. And so you might have been excused for thinking that she was cured, having had the melanoma 60 years before. 60 years of survival, no trouble, no evidence. You couldn't detect it anywhere, any scans, any clinical evidence of it, all gone. And yet this happens and she gets recurrence. So it is interesting that we may be assuming that all tumour has gone, every single cell, and maybe in some cases it has. But how would you know? Because small volumes of tumour might still sit there and are being controlled by the immune system. That's how powerful it is.
Yeah, but how would you know that the lady after 60 years doesn't have a new cancer that happens to be melanoma again? And she wasn't cured. Essentially, the definition could be anything anyone wants.
Yeah, it could. And she could be throwing off cancers all the time and these being knocked off by the immune system, and this one just got through. So that's quite true. It could have occurred that way. However, she had no evidence of any other melanoma at all. She had no evidence of any existing melanoma or ever having had it treated or burned off or anything like that. So, you can get melanomas that regress, melanomas that from no primary that you can identify, that they're all well recorded. So maybe that is the case. I don't know. But you could also hypothesise that with the lack of any other evidence of a melanoma, that this has sat there for 60 years and finally been activated. And we have other examples that similar things, maybe for not quite so long, but other examples of that too. So, it does raise a lot of questions about how we detect cancer, because clearly in small numbers of cells or single cell stage, you can't detect it. It's very, very hard to detect unless it's, you get lucky and just happen to sample that particular area. But it raises the likely scenario that, in fact, the immune system's constantly monitoring for malignant cells, and it's constantly monitoring for cells that are damaged or aberrant or injured and removing them. And so if this system gets disturbed in any way, then it opens the door for these cells to actually grow. And when they start growing, they start altering and become heterogeneous and start genetically mutating. And then you've got this collection of cells that are all of different types with different environmental evolutionary advantages too. So some cells are going to be more resistant to chemo, more resistant to radiation treatment, more resistant to the immune system. And what you really need is some sort of way of immunizing against this. And it raises a question of being able to vaccinate. The body probably vaccinates itself all the time. Almost certainly it does, because we are losing cells out of tumours. They're dying naturally as part of the tumour growth, so growth and death. And they're releasing components of those cells, and they're actually, the immune system's picking those up and then reacting. So there's this constant vaccination process going on in almost every cancer patient, almost certainly. And the challenge really is to try and harness that and to try and get a vaccine that can work against the individual tumours and also maybe malignancy in general.
Yeah, what stage are you guys at with this? Is it through clinical trials? Is it in clinic or not yet?
So what we've done is we've had the results of a moderate-sized clinical trial. We've treated patients with this type of vaccine with advanced melanoma, so melanoma that's spread, that's unable to be removed surgically. And then we've observed to see what sort of effects it's had. And we've observed some of these patients for, as I was saying before, over 20 years. So we really know that they can gain long-term survival if we get a really good effect. And what we've seen is that in 17%, we can actually remove all tumour, so we can get complete responses, which is remarkably high. We were very surprised by that because even in this major immunotherapies that have been approved already, the rate of complete regressions are only around 12% or so. You can get some other responses that allow you to live with the cancer and it's stable and so on, and they're excellent. They're of great value, but it sits at around 30% of cases for a lot of the single immunotherapies, and that leaves about 70% or so of people that actually end up dying of their disease. So this is what I was saying earlier too, that there's a massive area of unmet need where people really don't have effective treatments that they can go on with. So,
well, it may not be the only metric there may not be one important metric only. So 17% appear to go into complete remission, I guess I'll call it. But even if there was none, but the death rate was lower and people just lived with it as a chronic type thing, that would be a huge improvement too. So like, which of the metrics in terms of efficacy do you think is the most impressive, or which is included?
Well, the complete responses are the most impressive, without any doubt. If you get rid of all tumour, then that's spectacular. And it's something that I think should be the goal, in that patients really, desire that. And I think that sure, you can get, what we were able to generate too was a whole range of partial responses, which sat at around 20, just over 20% mark, and where part of the tumour reduced in size, and you could actually get partial regression but not complete. And then we had another percentage which pushed it up to the total. These were stable responses where the tumour just wouldn't grow, it just stayed put, which is effective too, and in a clinical sense. And so we had 78% of our patients had some form of clinically useful response. So that is the complete responses added to the partial responses added to the stable disease, which is reasonably high, which shows that this type of technique it's possible to actually make it quite effective in the repertoire of treatment of melanoma and possibly other cancers, because we now know that other cancers demonstrate similar sorts of molecules on their surface, similar types of molecules that their immune systems react to. So patients' immune systems react to. So you can actually have some activity of similar types of vaccines in other cancer types, I'm sure. We're yet to show that in a clinical trial, so our clinical trials have been restricted to metastatic melanoma, but we've got some other evidence that it might well work in other cancer types.
Well, excellent. So, Brendon, where can people keep tabs on this so that the moment that it's available or they can sign up for a clinical trial, they can do that?
Yeah, so what we've done is they can contact me through Adelaide University email. We've set up a company to try and sort of move this on a bit, because we need, clearly this is incredibly expensive and we need a lot more backing behind it to move it on, a lot more research to be done. So that's CanCuraVax, CanCuraVax.com. And we're hoping to start up a clinical trial fairly soon, looking at this type of vaccine in other cancers as well.
Well, very good. Yeah, no, that's excellent. I mean, it points to a whole new set of possibilities for many cancers, so it's excellent.
Sorry, Richard, I think this is a technology that sits right between cellular therapy, like CAR T cells and other types of cellular therapy, TIL cell therapies and so forth, on the one side, and then on the other side is the antibody type molecule therapies. So this sits right in the centre, this type of cell lysate type therapy. It's got its roots, quite a number of decades ago, but the technology has never been properly explored. So this is what we're doing at the moment.
Excellent. Very good. Well, Brendon, thank you for coming again on the podcast. And, it's good to have you in at least every 6 months for updates on this exciting stuff. Very, very important. So thank you.
Thanks, Richard.
Don't forget, before you go, use code GENIUS at Viome.com for an additional $20 off your health intelligence test, and get started on your health journey with the right foods, supplements, and probiotics and prebiotics for your unique biology. Get a deeper look within with Viome's health intelligence test. Viome, you decoded.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at FindingGeniusPodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.